So it says analyze why the sequence is poor quality and what you can do to fix it. So if this is the case, again, there's a video that Dr. Rashan put on the website that you can watch. But what could we do if we had an example of something like this, where we just have a repetitive sequence? We might not be able to fix that. We could try to come in from the opposite direction, maybe, instead of not hitting the repeat section. But if you go in the opposite direction, if you sequence in reverse, remember that you have to get through the poly A tail, because that's at the end. So that's another repetitive sequence you can't really avoid. It's one of the big reasons why when we used to sequence in reverse, a lot of our sequences were much more poor and they were poorer quality. In another case, if let's say you know that it's because the poor sequence data was because maybe there wasn't enough DNA, what could you do after that? Well, could we go back to the colony and regrow it and try to extract good DNA and process it again? We could do that. That's a little expensive because now we're repeating our work, but it could be possible that you could go back and go to the colony and regrow it. And the last one is, did the other runs go well? Did everything else get sequenced correctly at the facility? Could it be that the facility where you send your samples, or you sent your samples, actually had a problem or had errors? That's another question. You would have to contact the facility or Dr. Rashan and ask them, did this happen to anything else? And so it could always be the facility that they're using to sequence. But most likely it's us. Okay, if it's bad sequence, you're going to enter that as unreadable, and you're going to give a short description, poor quality waveform, or in the you could put in whatever description you want there. And once it says this, congratulations, you're going to submit. Now you just have to wait for someone to analyze it at the university. Okay, if the sequence is of good quality, then we have a little bit of work to do. Now we have to figure out, where does the sequence start? Where does the DNA start, or the gene start? And in this question, you can see that we're going to select yes. And then the first question is, in the unedited sequence, at which base does the cDNA insert start? Okay, so we know that this is our P-triple-EX2 vector, and this is our map that I've given you in class. I know I've told you that the forward sequence is what we use to start the sequencing reaction. So we have this area, this repeat section, and it's short enough that it doesn't usually cause a problem. And then you get past this area and you enter an area where you have an EcoR1 site. So you should look for, on your sequence, GAATTC, and then later beyond that, the ATTA. Once you see that, then you should have the following bases. And this is where it becomes different for everyone. It could have 3 Gs, it could have 4, it could have 5. The process where they inserted the gene into the vector puts in a different amount of Gs in this location. So this is the run of Gs, where after the last G, that's where you're going to start your insert. And that's where the sequence would start. Okay, so let's take a look at this. Look at regions on the waveform that match the polylinker region, which is what I was just saying. So we found the SMA1 site, and in this one, and we find the GAATTC. Here's our ATTA, and here's our run of Gs. Our insert would start right here at A55. Okay, so that's what you would be looking for in your sequences. And again, we've talked about this a little bit when I talked about sequencing. So this is the same process we showed before. So at the start of this clone, we're going to put A55 in this spot, because that's what we determined it to be, the start. Now, your first question on your quiz. Take a look at this sequence, and there's your map. And tell me where the start of the sequence is, or the unknown gene. Number 2, at which base is the start of the forward insert? Again, you're looking at the map and looking for the run of G's. Which base is a start for this forward insert? And which is the base of the start of the forward insert here? Okay, so this question, number 5, which is the proper answer for the start or quality of the sequence? Here you have an example where something went wrong. So where would you call the start, or is it a bad sequence? Okay, so how do you determine the endpoint of your sequence? If you have a small insert, then most likely the polymerases made it through the entire sample and you see a poly A tail. If you have a medium insert, it becomes questionable as to whether or not the polymerase is going to get there. And so if it doesn't reach the end of the poly A tail, you have to make a call of when you think the end of the sequence is. And you want to cut it off where it starts looking very poor, but go as far as you can before you make that call. Okay, so again, here's the end of the sequence. We look for the poly A tail, and even though this has a little blip of a C here, I'm going to end it right before because I don't want to, that's definitely not part of the poly A tail. What if I don't find the poly A tail? Again, you go until it's unreadable or becomes unreliable. And here we are entering A765 into our form in Desub, and you're going to then what I normally do is I highlight the rest of the sequence, I delete it, and then I go to the beginning and I highlight the beginning of the sequence and I delete it so that I can select all and paste it in here. Okay, and again, here are some more questions for you to be able to identify the end of the sequence. Okay. So in this case, you highlight the desired base pairs for the insert. Okay, delete it, like I said, highlight the 5' and delete it, and then you paste it into this box and you hit next. And basically that's where you end cropping the sequence. At that point now, we're going to go on and take a look at ways to analyze it using tools in NCBI. So hopefully this was helpful, and we will see you in class. Have a nice day.